Here you go.
My project was on bioremediation. My group's research question was how copper and zinc affect the growth of the Brassica neriga plant, and my question is important because and interesting because we will get to find out if zinc and copper can save the plants from if there was an oil spill or something like that. My group used zinc and copper as the two main things that were used in the plant to see as the toxins. I learned about the structure of the plant cell and that the cell is a basic unit of life in plant cells, unlike animal cells. Also that plant cells are eukaryotic cells and that they differ from several key aspects from the cells of other eukaryotic organisms. Also that I learned about photosynthesis, and that's the process of plants making food, and that also bioremediation at the cell level is that that they're restoring balance, and that it can use it wherever the water environment has become polluted or is suffering from ongoing pollution. Our hypothesis was that, my group's hypothesis was that the plant with zinc would grow better than the one with copper. Our variables were one independent variables was that the zinc and copper, sun, water, and food. The dependent one was that the height, and control was the amount of copper and zinc, amount of dirt, and amount of water that you can use. Our process was that you take 2 cups and a pen and a string, and you poke a small hole in the bottom of the bigger cup. You put another bigger cup and put water in it. You put dirt in the small cup and seeds. Put the smaller cup over the big cup, put some water in the top of the cup, also in the bottom, put zinc and copper in separate plants, and water every Thursday, and make sure to take measurements. My graph was, this was the data table that every day each plant, the current height, also the average height, and average change in height, and we also put some notes at the bottom and kept track. Our qualitative observations were that the copper plant grew really fast and then started dying. The plant that we put zinc into started off short, but then grew taller in the end. And our graph, as you can see, has like the blue was the one with the zinc, and it started off really small and like teeny, and then it shot up, and at one point on the date, close to the date of 9/29, the copper and zinc were close to the same height. And then the copper shot up at that point, then kind of had like a steady growth, then shot up again. But the zinc had a steady growth for a moment, then around 10:20, 10:13 in the middle of that, it kind of slightly grew. Our analyst was that we saw trends. The zinc stayed completely steady around 9:25. Both plants were at 5 centimeters. They both shot up later, probably because of the effects of copper were wearing off. Our comparisons were that the copper started out strong, stayed steady, and shot up a bit at the end, getting up to 13.2 centimeters at the end. The zinc only got up to about 7, therefore our hypothesis was wrong. Our conclusion was that the data does not support our hypothesis because the zinc did not grow better than the copper. The copper was the one that was better in the long run. From what I see in the data, all the chemicals were wearing them down, and they couldn't grow. Once it was later in the growing process, the chemicals kind of wore off and the plants were able to grow. Bioremediation in the real world. It means that you have to wait a while for the effects of the chemical to wear off. The effects were pretty weak. They were still recommendations to Watertown is that this is a good possibility for help. It's not going to solve your problems, but it's going to help all the effects of the chemicals. What would you do? What I would do differently would take more data so I could find out the small details easier. And my recommendation to scientists is take data every Thursday, because that's what we did, and watch your plant change its colors, because ours did, and we didn't really watch that too much. And my sources. Yeah. The end.